Welcome to my talk, The Denizens of the Dead Zoo. I'm sure that most of you know that the Dead Zoo is a friendly name that we have for the Natural History Museum, which is over on Merrion Street. We try not to refer to the whole of the natural history collections using that term because fundamentally it's not a dead space. It's a space where a lot of things are going on and there's a lot of life there. So that's kind of what I'm going to be talking to you today about. But I had to start with this beautiful specimen from the collections, which was on display until recently. He's suffering from a similar problem to me. He's balding, and it gives him a rather more threatening appearance than he had before he started losing his hair. So these small primates, these tarsiers and their relatives are they're charismatic little things when you see them in the flesh. They don't make good pets. They really don't. So if you see pictures on social media and so on of people having them, it's really bad. It's one of the things which we try to encourage people to avoid doing is getting exotic pets. It's a really bad idea. It's bad for the animals, it's bad for the environment, it's bad for you. So the Dead Zoo has fondly been referred to as a stately home of death. Not by one of its detractors, but by my predecessor, Nigel Monaghan, who was the keeper of natural history before me, and it was used as a term of endearment. When you go to the museum, you can't go at the moment because we're closed, and we will be for a few years, because we're getting major redevelopment. But I'll show you some pictures. If you're not familiar with the space, it does have that stately home feel with lots of game heads around the walls and an amazing Victorian style cabinet museum. So it's quite evocative of the past, and it's often referred to as a museum of the museum. So one of the things that space was trying to do was to give across a sense of not just what was out there in the natural world, but to illustrate that. So this is one of our, this is just for the spooky effect, we are coming for Halloween. This is a bird-eating spider with a hummingbird. This is currently on display in the Dead Zoo Lab, which is over in Collins Barracks now. We've opened that space. It's not really, I won't refer to it as an exhibition because basically we put some things in some cases and we're experimenting with how we display them. And the idea is that we will talk to our audiences and get a better idea of how we might redisplay things in Merrion Street when we reopen. So the idea is it's an experimental space. That's what we call it, the lab. So that's basically one of our cool specimens. In a way, the whole point of the Dead Zoo was to historically show people things which they just wouldn't have ever seen. In the days before David Attenborough and Wikipedia and all those sorts of things, the best way to see what was out there in the world, the best way to understand biodiversity, was to see this huge diversity that you'd get displayed within the museum walls. It's almost impossible to see that kind of diversity if you go to the zoo, or even actually if you go on safari or whatever. You often don't see anything like what you'll see jammed into that small building, and it isn't as big as you'd expect. And it hasn't really changed very much over the years. So this is a photograph taken in 1910. This is a photograph taken just before we started to empty the building for a decade. So just to put that into perspective, the lighting has changed. We shut out some of the windows, and actually, that's a little bit disingenuous. A lot of things have changed in that space, but fundamentally, it looks and feels much as it did before. This is very much the kind of the forward, public-facing bit of the museum. We had around 10,000 objects on display in that space. That is the tip of the iceberg. We have over 2 million objects in the collection, and those collections get used really, really actively. They're historic. So this is an example of. Again, it's one of the nice spooky kind of Halloween-y type photos, but this is a large octopus from New Zealand, and each of the specimens we hold contains really interesting information. So this is a paper which was published on this particular octopus specimen, the well-travelled octopus from Dunedin to Dublin in 1886. The whole point of these collections is that they can tell stories not just about the animals, but also about trade, about the way in which people interacted in the past and how networks of science were created, developed, and basically share knowledge around the world. And there are colonial legacies which are associated with that, which we can go into. But fundamentally, they talk about all sorts of things. And this particular octopus specimen is really unusual in that it's preserved using a technique which is very, very rarely used, and people don't really understand how it works very well, because it was done just on a handful of specimens, it's glycerin preservation, hot glycerin preservation, and preserving octopus flesh is actually quite difficult. You can pickle it, but then it loses its colour and it doesn't really look the same as it does when it's alive. In this instance, this hot glycerin method was impregnating the skin or the flesh of the octopus with glycerin in order to preserve it in its kind of more naturalistic position. Understanding these sorts of preservation techniques are fundamental to us because they mean that we know how to look after the collections going forwards, because if you don't know how it's been prepared, you can't really look after it. But also, we have we also have to look after them going forwards, and we need to be able to tell researchers what's in there. So if people are doing, for example, genetic work on one of our specimens, we need to be able to tell them if it's been impregnated with something which may influence their results. So this is actually supposed to be an animation. This is a specimen of a frog, and one of the big issues we have is that we have around between 250,000 and 1/3 fluid preserved specimens. That's a huge amount of material, a huge amount of alcohol. We preserve everything in around 70% industrial methylated spirits. Ethanol is better, but it's a lot more expensive because you can drink it, and then you have to pay duty on it. So we avoid using that, except for specimens which are specifically going to be used for DNA work. And I'll talk about that in a while. But this is an example of what you can actually do to recover a damaged specimen. So this is actually the after picture from this. Okay, so it looks like that's irretrievable, but actually using good techniques and understanding the conservation processes behind it, you can actually bring it back to life. This is, if you ever want to read about it, there's a blog post here. Sorry, the formatting on my slides has got a little bit messed up by the transition to this computer. There's the Natural Sciences Collections Association is a really great organisation where we share practice. If you go to the NATSCA blog, there's actually a full detailed list of like how you do this, with loads of photos and stuff. So if you're interested in getting a dried out frog and turning it into something that looks like a living frog, you can do it using those techniques. Okay, so oh, there you go. There's that's halfway through the process, just to show that I haven't just put two frogs side by side. This is the midfield during that process. So understanding how all of this works is really important for us because we have to look after these collections. And a part of that is actually looking after the building, that everything's in, and making it fit for purpose. That's as it was, that's how it is now. We have emptied it, it's completely empty. As part of that process, we removed all of those things that make it a stately home of death. Maybe not all of them, but most of them. And this is kind of what you think about a lot of the time when you think of the Dead Zoo, the Natural History Museum. You know, 333 game heads around the sides. It's what jumps to mind. What you don't think about are all the tiny things in the collections. So this is a lovely comparative example of wasp heads. Very useful for identification, and we need to add to this because more wasps are turning up, hornets and so on. And I'll talk about those in a bit more detail later. But fundamentally, the collections we hold aren't just there for that kind of public engagement piece. Museums have a fundamental mission to help understand the life of the planet for the benefit of the Earth and its inhabitants. This is a really good paper talking in detail about the impact that natural science collections have on our understanding of biodiversity and things like climate change. And so I'm going to talk a little bit more about that as well. So just to give you an idea of what goes on behind the scenes, when you're not looking at all the kind of nice Victorian cabinetry full of dead stuff, what we're talking about are specimens coming in all the time. Species new to Ireland, so this is an example of a couple of bee species which turned up, they were collected in 2015, 2017, new to Ireland, got lodged with the collections. These are flies, also collected in probably about a 5-year period and lodged in the collections. And actually, that's half of the number that came in. All of them are new species to Ireland. Okay. This is one I actually collected when I was on holiday last week. It's a field vole. They are an invasive species which have turned up in Ireland recently. And again, we know about that specimen because someone contacted us. It was killed by a cat. I identified it. We wrote up a little paper just to say these guys are in Ireland. And now that's being built on, and we are finding more and more examples of this species turning up. Okay. This is actually from an owl pellet. And the reason I've got this photo is because this little cusp on this tooth here is how you can tell it's a field vole. Otherwise, it's really hard to tell it from a bank vole, which were already in Ireland, been around for around 100 years or so. Although this is from Cavan, and I don't think the bank voles have made it over to Cavan yet. So other species which have turned up new to Ireland, these are all just from 2024. This one here is this ant-like flower beetle. It has this cool little like tusk almost on its thorax, and that gives it this name, Monoceros, which just means one horn. So this is a species which is first recorded on some flowers in County Kerry, and oh, that's really not worked very well. That's but basically, those are the sorts of specimens which are collected and put into the collections. You know, so people go out and collect things. You know, people have a fascinating interest in what's out there. This guy Edwin Bullock, who you can't see because he's been squished, really squished. Even his name has been squished. Edwin was a very prolific collector. He was based around Killarney, in what is now the National Park. It wasn't when he was collecting. And he went out every day for about 50 years and collected. He collected everything. A huge diversity. We have these all Bullock specimens. You can see there where they've got these red labels. These are types, and I'll talk more about types in a little while, but they're really important specimens. And he basically didn't have enough space to store all of these things nice and neatly. So he had to use this multi-story system. So you have multiple specimens on one pin. And when you start considering that there are drawers and drawers and drawers of this in cabinets of these, and we have multiple collectors feeding into that kind of collecting process, it means we have this incredible repository of collections from Ireland, which basically provide us with a baseline understanding of the biodiversity that we have. And it's constantly growing, but also we can use it to compare change. So the tawny mining bee was considered extinct about 100 years, and then it turned up in 2012 in Kenny, and it's been turning up in other places since. And the thing is that we know that it was present in Ireland because over 100 years ago, we had them in the collection, collected in kind of the 1890s. That's one of the reasons that collections are so important. They allow us to provide a sense of what has changed. So it's not just about what's new in and what's here now. It's about how that has changed over time and how that can be impacted by things like climate change or farming practices. So the collections serve a really important function to understand change. And really the most important part of that is to feed the data that we've got from the collections back into other places. So the National Biodiversity Data Centre collates the information. So they go out and do monitoring of what species are out there today, but also they will take the records from the collections and put them onto the database. And that all gets fed up to GBIF, the Global Biodiversity Information Facility. So anyone in the world can find the information about a particular species and its distribution globally almost instantly. It's a really, really useful facility. And these are the sorts of projects, digitization and making things available online, they're really, really fundamental. And again, I'm going to talk more about those in a second. So just as these are the kind of species distribution maps that can be generated by having just that locality information. And a huge amount of what we know about Irish biodiversity today is actually based on the published checklists from the museum's holdings. So Jim O'Connor, who was the entomologist before our current entomologist, Aidan O'Hanlon, he published literally hundreds and hundreds and hundreds of checklists and a huge amount of really, really important information about what species turn up where in Ireland. Incredibly valuable, and over time it's getting fed into these digital systems. And one of the things about the collections is it allows us to see occurrences when they're not necessarily actually living in Ireland. So this is an example of a carpenter ant from North America, New York carpenter ant. And this is actually a specimen from our collections, which came in around 1930. We haven't seen it since, hasn't turned up. It came in on lumber, but knowing that it can turn up in that way means that people be more vigilant about it and actually take steps to try to reduce the risk of what could potentially become established as an invasive species, actually setting root by controlling the way in which things come in. So if you know that there's a risk of something like carpenter ants turning up, you can take steps to freeze or heat treat or whatever it may be, the timber that's coming in. This particular timber shipment came from Canada. But if you don't take those steps, you can't control it. Yeah.
Are carpenter ants dangerous?
They're not dangerous, but they can cause damage to wooden structures. So that can be one of the issues. So a big one at the moment is, and you've probably heard about this, is the Asian hornet. So entomology curator Dr. Aidan O'Hanlon has been really, really active in trying to get to grips with the presence of this species in Ireland, to help lead out on training members of the National Parks and Wildlife Service to be able to identify them, track them, get their nests. And actually that's one of the things which we recently got. So there's this is the nest that was taken in County Cork, actually in Cork, by the NPWS, and we have that in our collections facility now. We also have the nest from Cove, and we're dissecting those out because one of the things which you need to understand is, in order to control that population of Asian hornets, is where are they in their breeding cycle. And so that's why it's really important to actually dissect these nests when they're taken, because that will tell you where they are, how many mature adults you're expecting to find, how many larvae, how many queens might be getting produced by that nest. Those things are fundamental to controlling that invasive population. And what we do know about the Asian hornet and where it's coming from is largely based on this specimen, which was taken in 2017, I think that came in. And it was used for DNA analysis. Because we had it in the collections, it was actually found in Dublin. It was one individual. Everyone was a little bit unsure about whether it was from a population or if it had just turned up, like from the airport on someone's luggage, whatever it might be. So one of the things that we did was to make one of the legs of this specimen available to be used for DNA analysis in order to understand where it came from. It turns out it came from a population in France, which is the same population which has also come into the UK. So it seems likely that it's actually one that just made it over, which is worrying. And actually that probably is what first started sounding the alarm bells that the Asian hornet might become really well established in Ireland really quickly. And it's one of the things that we've seen with other species like box tree moth and things like that. You see a record turning up in around 2017, 2018, and I mean now box tree moth are everywhere. Certainly in Dublin, they are like decimating the hedges. And these sorts of problems are, it's very easy to kind of overestimate or underestimate how much damage can be done by an invasive species, the impact that they can have. Other things that come in. With climate change, you have things like changes in the insect vector species on nocturnal. So various mosquito species, midge species, biting insects. And obviously biting insects can carry disease. So when you think of things like dengue fever, malaria in particular, everyone's familiar with malaria. So if you start to see changes in biting insects that turn up in Ireland, then you need to start planning. So one of the projects which has been running through the Health Service Executive, coordinated by UCD and with active involvement from us. So this is our little, this is actually a mosquito breeding chamber. So you get the mosquito larvae or the midge larvae, you put them into this manky water, and they do what they do, which is grow into adults. And then when they emerge, they fly up here, get trapped in here, and then you can kill them and identify them. But it's really hard to identify them from the larval stage. Now, some people are very good and can identify them just from the exudia, which is the shed kind of casings from the larvae. But that's a really, really specialist skill, and only a handful of people can do that. Whereas you can identify the adults much more easily. So this is quite an effective way of breeding them up and actually identifying what problems you might be facing in the future when you understand what species are present today and how that can change. And yeah,
Examination, tiger mosquito there.
Yes.
And they're in Ireland?
There have been occurrences, yeah. Not many, but there have been some occurrences. And this is one of the reasons we're worried. And this is why there's actually quite a lot of work that goes on behind the scenes. So NetVec is the name of that project. And it works across several of the universities, the health service and the museum to try to get a handle on which vector species might be turning up and becoming established. And I think the big issue is turning up is one thing, but getting established is another, and that's what you need to be really concerned about. And obviously there are things that you have to do in order to manage that, whether it's changing the way in which you deal with your waste, so you don't get pooling water. There are multiple ways of dealing with these problems. But it's really important because fundamentally, if you get a new infectious disease turning up in Ireland that you're not kind of geared up to deal with, it changes everything. It changes agriculture, it changes the way in which you manage your animals, it changes human health. It means that suddenly, you might need to sleep with a mosquito net. In a country where that was not been a problem for a very, very long time. There have been some warm patches in the past where it may have been, but it's not at the moment. And we're not used to it. There's nothing set up to be able to cope with it easily and quickly. Emerging diseases are hugely problematic. We've been through a pandemic fairly recently in the last 5 years. And there are constantly new diseases emerging, many of them with a zoonotic origin, so they come from an animal of some sort, and it goes over that species barrier. There's a really interesting piece of research done in 2010, so it's quite a while ago now, looking at bats in India. They identified over 55 viruses in the bats that they sampled, and 50 of those were new to science. When you consider that COVID, who have kind of come to the conclusion that it was almost certainly bats, either directly or by some intermediate means, bats ended up being the main contributors to the COVID outbreak. It kind of gives you an idea of why it's actually quite useful to study these bats, which are held in collections, where you can still extract DNA, you can still extract RNA, you can still look at what loads are going on there to get a better sense of which species might be out there. Viral species, so that you can start getting a step ahead of any outbreak that might happen in the future. Because if you don't understand the physiology, the anatomy of a virus, you can't do much to actually try to address the problem of it becoming widespread. And so this is actually there's a paper about this as well. Again, it's one of these ones which got squished. I don't know why, but it's got squished. But it's a great paper, and it's really just talk about how collections should play a much bigger role in predicting future outbreaks of diseases. Because effectively, they're cheap. You can go and walk in, you can get some stuff out, you can go and sample it. You don't have to go to India or to, I don't know, anywhere in the world. You don't have to go to China to go and get the data. You can literally go to a museum down the road. But people don't like doing that compared to going somewhere exotic and nice.
When you say you review the DNA, can you check as well if they have viruses as well?
Yeah. You can check for RNA as well. Yes. Yeah. And it's a missed opportunity. Voucher specimens are really important because just moving kind of away from the disease aspect, the voucher specimens which we collect, whether it's mosquitoes or bats or whatever, they aren't just kind of a record of that species, because they give you that repository of data. Whether it's viruses, the DNA of the actual animals, the DNA of parasites that are associated with the animal, they play a fundamental role because you've got a physical, tangible representation of that thing and the things associated with it. So that's why we still take voucher specimens. So we have, for example, this is a voucher specimen, well, not really a voucher, but it's just a collected specimen of a sunfish from government collections. Mostly used. Yeah.
Sorry, I didn't quite catch. What's a voucher specimen?
Oh, sorry, a voucher specimen. So a voucher specimen is literally an example of what you've collected when you've been doing this field work. So if you go out and you're sampling for mosquitoes, for example, or bats, and you're doing some survey work on diseases they might carry, you take some samples of that species so that you can take that back and actually have it there preserved in the museum. So if anyone comes along later and says, well, I actually don't think this is the species that you think it is, because a taxonomist has come along and said, actually, there are two species there, or a species complex. We don't know that you're talking about the same thing. By having the physical voucher specimen, it vouches for that piece of data that you've published. So it's really important having voucher specimens.
Sorry, did you say what species of bat was responsible for 55 cases?
It was, I can't remember which one it was actually.
What?
I can't remember which one it was.
Okay.
I'll try to find that out.
It was in Ireland?
No. It was in India.
Oh, right. Okay, thanks.
Yeah. So this is an example. This is a sunfish, one of the biggest bony fishes on the planet, actually. Huge, great things. Awkward to move. Awkward to display, actually. This one's so big that it was going to squeeze into its display case and was all, like, the fins are all kind of squished up like that. They're really cool fish. They feed on jellyfish. They swim around in the oceans. They're called sunfish because they like basking in the sun. They turn to the side and present that large area. They are kind of weird. They don't have a swim bladder. They use blubber, basically, or fats as a way of staying kind of in the water column at the right level. And they're quite unusual to find because they tend to hang around in the ocean. People don't really catch them commercially. And so you don't get many specimens turning up. So recently one was stranded down in Donegal. And this is us with a team of very helpful chaps with a truck because they weigh, that's 200 kilos, that one. So we're getting that for the collection. It's currently in a freezer in Athlone and it will be dissected. We're going to prepare a skeleton from it because we already have a taxidermy example. And what we'll do is investigate the stomach contents as part of the process and take various samples from it, tissues and so on, because all of those things can give us information. So, for example, they feed on jellyfish, and we find that a lot of species that feed on jellyfish are heavily affected by plastics in the water column. So we want to see if there is a larger percentage of plastic in the gut, for example, than you might expect, and help to understand why it's stranded in the first place, because sometimes it will be that their guts are so packed with plastic that they behave differently, and that puts them into positions where they can strand. And actually, you know, that whole thing about the one specimen not just being one specimen, these are parasites from sunfish. So when you take a specimen, you've also got the parasites associated with it. We actually had a researcher in recently who was going through our thylacine specimens, the Tasmanian wolf, which is extinct, went extinct in 1936. Well, probably went extinct a little bit after. The last known animal died in 1936 in Hobart Zoo. And we had someone come in to sample it for parasites, because no one's really looked at the parasites of them before. And they found parasites. Even on a taxidermy specimen, they went through it literally with a fine-tooth comb and managed to identify parasites which had been living on the animal when the animal was alive. These are parasites of the species that is extinct. The chances are, because parasites tend to be so host-specific, that is probably also an extinct species. And you would never know that it existed in the first place if we didn't have museum collections to be able to provide that kind of data. So tracking change is a big part of what all those collections are for. And you know, this is going back to that field vole example I was telling you earlier. You know, we hold comparative material to help people make identifications. So when we published the paper talking about the presence of a new vole in Ireland, we used this image here and this image here because they help to show people the difference between the short-tailed field vole, which has a short tail, and the bank bulge, has a shortish tail, but not as short as the short-tailed field vole. So they're quite hard to tell apart. I mean, they do look pretty much the same, and you know, there can be a lot of variation between individuals within a species as well. So this is just to give you a few clues about how you can differentiate between the two species. And here, you know, I mentioned about the little that little cusp on the tooth there. So you've got this is a more worn tooth from an older individual, which is why it looks a bit more different to that. It doesn't look as jaggedy. This is the bank vole, and it's not got that extra little cusp there. So you've got 1, 2, 3, 4, 5. This one's got 1, 2, 3, 4. These sorts of details taken from things like, you find them frequently in owl pellets in particular, but you can find them in other places too. Cat kill is very common as well. They can tell us what is going on out there, but you do need to have that kind of attention to detail, and having the museum collections there for comparison is hugely valuable. So really, I mean, I'm talking about, you know, we do research in the museum, but mostly we make our collections available for other people to do the research. And there's some really cool stuff going on. You know, whether there's like taking a fine-tooth comb to thylacines, or they're looking at the genetics of bats, or whatever it may be. So, you know, previous speaker Emma Teeling, you know, she's used our collections in the past quite extensively. It's one of those things which, you know, it's a resource for everyone, not just for scientists as well. We actually, you know, artists come and use collections all the time as well. But one of the big issues that we, as kind of an organisation, are keen to do is to help support this idea of understanding what we have out there, and mainly supporting taxonomy. So taxonomy is how we describe what things are, give them names, organize them and give them names. And while that may sound really kind of boring, it's only boring if you don't do it. Taxonomists get so excited about this process. It's a cool process, and you get to give things names, which is always fun. But also you get to discover something new, which is, it's like going to the moon, or how often do you discover something new that is fundamental to our understanding of life on this planet? It's rare. And the whole point of taxonomy is that you're setting an agreed common frame of reference to recognize a species, so that everyone's talking about the same thing when you say a particular species. We all know what Homo sapiens looks like, more or less. And if you've seen a dog, they look quite varied, but usually you can figure out a dog's a dog. When you start dealing with some of these species that you don't see on a regular basis, it can be really difficult to identify them. And that's why having those specimens there, which have been really thoroughly described, is fundamental. Because if you can't name it, you can't protect it. And that is literally, so for example, if you don't have a formal scientific description of a species, telling people how to identify it as the thing it is, you cannot put that into legislation. There's no way. So there's this piece of legislation called CITES, this Convention for the International Trade in Endangered Species. Really important piece of legislation to try to prevent things like poaching of elephants, reduce the ivory trade, and actually clamp down on a lot of that kind of illegal activity that leads to species going extinct. And fundamentally, it only really works because people can say what something is to give it that protection. Otherwise, it's not protected. So, and just to kind of go back to what I said before, new species are still being described in Ireland. It's not that it's a done deal. We tend to have this kind of a slightly arrogant attitude towards our understanding of our own place. It's like, oh, Ireland's small, it's got a relatively small biodiversity. Sure, we know all that. There's probably at least 2,000 species on this nation that have not yet been described. Okay? There's lots that still needs to be discovered, lots that still needs to be talked about and understood, described, and made available. This is a new species and genus, actually. So it is very, very, very underknown. That was described from Ireland very recently, from the north, but still, it's from the island of Ireland. The common name is the fence climber twiglet weevil, and you can kind of see why. And they like climbing fences, that's where they found. But they found quite extensively, and they turn up in Cornwall as well. So it's a species that has never been described. It turns up in Cornwall, in the UK, and in the north of Ireland, on fences, like where people are. And no one has described it until, I think it was last year. It gives you an idea of just how much there is still to know out there that hasn't been described. And that particular specimen is one that's lodged in this one in the museum? This one's in the collections in Northern Ireland, but we also have examples of this species which are parts of a type series, so that if people find it, they can come and compare it and they can know what they're looking at. It's really important to have these types. And these type series, this is a really, really cool octopus, Benetoctopus besticus. This we made available to researchers in Australia to be able to do taxonomic work, because they were studying that whole species complex. So literally, this is what I refer to as the devil's burrito. It's this octopus wrapped up in muslin and soaked in alcohol, and then we put that into heat seal it into a plastic bag, pack it carefully in a box with padding, and then shipped it to Australia, where they did the work and shipped it back. So, and in this particular instance, this one, something went wrong with the shipping. So it got to the UK, and then they didn't quite know what to do with it. So they sent it back to Australia, and then they had to send it back to us again. So it literally kind of went around the world twice to support the scientific research that goes on in taxonomy. And while it is essential that we can provide collections that can do that, it's much better if we can do it digitally. So this is an example. This particular specimen, you couldn't really send this very effectively. I mean, you can send these in the coast. And we have researchers in India, in Florida, in various parts of the world where we have sent these specimens to. This, just to give you a bit, that's 0.2 millimeters on that scale bar. This little tiny parasitic wasp is about the size of a full stop at Times New Roman 12 point. Okay? Just to give you an idea of how minuscule it is. And this photograph was taken by Elijah Towers, who's a taxonomist who works on this group. He has rigged together his own specialist equipment to be able to get photos of these tiny things, stack the images so that you can actually see in detail, because it's almost impossible to get a good photograph of one of these things without a really specialist setup. We don't have that kind of setup in the museum because, you know, I've got 4 curators, which is the best staffing that we've had in a very long time. Because when I started, there were 2 curators. So it kind of gives you an idea of what we do. And we've got 2 million objects to look after. So we don't really have the specialist equipment and the time and the resources to do this level of photography. But that's one of the reasons we open our doors to researchers. Elijah came to us. He stayed for a couple of weeks. He took photos of about 70 type specimens of parasitic wasp in the collection, which we have accessible now. We can send those to anyone in the world. And you know, that's the whole point of the taxonomic community. It's about collaboration. So this particular specimen, we've sent that image and various other ones which show all the taxonomic features to researchers all over the world. Hugely important resource. And that whole idea of getting the collections available online is fundamental to making taxonomy easier, making it more effective, and also just generally feeding into the scientific understanding of biodiversity. So this is an example of taxonomic resource. It used to be called the mollusca types in Britain, but now it's the mollusca types in Britain and Ireland, because between us and the National Museums of Northern Ireland, we've contributed around 300 type specimens of shells, mollusks, to this project. They came over, they did the research, huge amount of taxonomic research, took the photos, shared it globally on this website, freely available to anyone. Really, really valuable resource. And lots of other institutions in the UK have fed into this as well. So effectively what you have is a repository of type specimens from all over the world, because the British Empire wasn't just taking stuff from Britain and Ireland, they're taking stuff from everywhere. And it means you've got this hugely valuable resource for taxonomists working on these groups. Now, when you've got digitization, you also have the opportunity to do research using some things like computer vision. So you can automate a lot more processes. And so, again, if you're looking for signals that are hidden in collections and you have enough good quality data, you can start taking measurements, you can start doing things without actually having to do all the work yourself. So things like AI, well, you know, things like ChatGPT are really questionable at times, and you're like, oh, is this a good thing? Is this? Some of the really powerful AI systems which are analyzing data are hugely valuable, hugely important. They present massive opportunities because they can process things so much more rapidly and, I mean, not perfectly, but humans aren't perfect either. So what you can do is you can build up enough data to get a clearer idea of what's going on. And that's actually really important for understanding things like how climate change is impacting on morphology, which is what's going on with this particular piece of research here, looking at how you get asymmetry, fluctuating asymmetry is a big thing in biology. If you're interested in biology, you've probably heard about fluctuating asymmetry. But asymmetry can be driven by external forces. So as you get more climate stress, butterfly wings become more asymmetrical, and bees as well. And obviously, as pollinators, this can be really problematic if it does actually impact on their effectiveness, on their efficiency. This is an example of a project where the Signify project was a great project. I had to put this picture in just because this is like the signifying bird for the project, the Signify project, which is run by the Li Kongxian Museum of Natural History. Basically, we hold a small number of objects, a couple of hundred, maybe 250 specimens from Singapore, collected from rubber plantations, because rubber plantations were big business for a very long time, a lot of time really unethically as well. You know, I'm not going to go down that route today. So these group of fantastic people came over again for a couple of weeks, digitized all of the specimens from Singapore, really good quality imagery, beautiful images. But most importantly, they took the data, they took the images from every angle, and it means that they now have what we'd call a digitally repatriated collection, where effectively they have all the information. I mean, obviously not all the information because the specimens are still in Ireland, but a huge amount of information to help inform biodiversity work that's going on in Singapore today. And a very practical example of that is this 7-horned mud snake. So this is one of the rarest snakes that you get in the Lake Peninsula, and there are only 9 known records in the world across all time. So it's pretty scarce. It's hard to find them because of the environment they live in. And in Singapore, a specimen turned up in 2021, and they weren't sure whether it was a new species to Singapore, whether it had been there historically. And the only way they were able to figure that out is by talking to us, because we happen to have one of the specimens, this particular specimen, in our collection, which we know was collected from Singapore, from a rubber plantation in 1914. Is that right? Yeah. Fundamentally, this was intrinsic to understanding that you're looking at a species which is always present and has just not been collected, not been sampled, not been seen, rather than something new that's turning up. And that's really important, because if you don't understand your baseline biodiversity, you can't say how it's changing. And there's a nice book that came out that looking at some of the stories there. Another example of that is specimens collected by this guy here, William Thomas March, who was a free black man in Jamaica, who was an avid collector. He collected all sorts of plants and animals, sent them all around the world to contribute to the scientific knowledge of Jamaica. This is a type specimen of the Jamaican petrel, the blue mountain duck, as it used to be known. Just to give you an idea of how taxonomy has changed, we could never consider this to be a duck these days, but that was the terminology that was used when it was first collected. And that's actually really important, because changing our understanding of diversity and what things are is, it has to happen. That's what science is. It's about change. It's about understanding things better each time. It's not about getting the right answer. It's about getting an answer which gets you closer to the truth, or gets you closer to a full understanding. And it's an iterative process. You can only ever build on it if you keep sharing the information and challenging it. So that means that when people in Jamaica are doing research, they will often contact us because we have specimens which that March collected, he sent to Ireland, we have them here, and they will say things like, we're not sure if you misidentified this. Is this a least tern, which is a common species, and you find them everywhere, but in this kind of report that he wrote about the species that are present, he said it's a different species. Could you just check to see if this species, which we've never found on Jamaica, but it says it's in your collections, could you check the identification on it? And of course we can, and that's what we do. That's what I was doing here, checking the identification. It turns out it was just a least term. So it's another example of how taking that voucher specimen effectively means that you can check an identification, and just literally by that one piece of work, you know whether something's gone extinct or was never there in the first place. If someone recorded it as being there and they misidentified it, and then you've never seen it since, how do you know if it's gone extinct or if it's just misidentified? You have a voucher specimen. You can go back to it and check it. And these are the sorts of things that happen on a regular basis. Now, sometimes things have gone extinct. This is an example of the Auckland merganser. This is a really cool little duck. This is actually a duck. And this is from, if you can guess, from Auckland, New Zealand. And the last one was shot in, I think it was actually I think this might be from the last series that was shot, 1901. This guy, Lord Ranfurley, I think he shot all of them, pretty much. He was, I don't know if he had something against them, but he loved shooting them. He shot a lot of them, and there aren't many in collections. I think there are 7 remaining specimens. I think there are 3 taxidermy, and the rest are fluid preserved specimens. And it means that there's an entire lineage of birds which we know relatively less about because they've been more extinct. And actually, this specimen and others have been sampled for DNA to get a better understanding of how the merganser species complex has distributed itself around the world. That tells us about evolution. It tells us about all sorts of interesting questions about adapting to new environments because it was flightless. So these are the sorts of things that you can unpick from the DNA, still preserved in these collections. This is shot in 1901. Still got good enough DNA to get a sequence, a full sequence from it, because the techniques for extracting ancient DNA have led to museum specimens just opening up a whole new area of research. And this is sort of a fairly old quote from Dr. Michael Rossello, who was working on the Galapagos giant tortoises. And he's just pointing out that as the biodiversity gets worse, museum collections will become more important as a repository of genetic information to help understand species conservation efforts going forward into the future. And in Ireland, the stoats, the unique Irish stoat is one thing. We also have been supporting research into nightjars. And there's a paper published, I think it was earlier this year, talking about nightjar populations and reintroductions potentially into Ireland, because they're pretty much gone. But understanding the genetics of the population that used to be here can help inform a good option for reintroductions if you do want to go down that route, because you want to find one which is as close to one that used to be here as possible to make sure it's got as much in common with the ones which were lost as possible, because it'll be better adapted to that environment. That's the thinking. In terms of genetic resources, a massive one we have is the Irish Cetacean Genetic Tissue Bank, which is basically a citizen science project where people all around the coast of Ireland, north, south, everywhere, collect little samples of skin from stranded whales. So whales strand on the shores around Ireland all the time. The Irish Whale and Dolphin Group do a huge amount of work to capture reported strandings. If the animal's still alive, they try to refloat it. But that's very rarely successful, very rarely successful. Most of the time the animal dies. And so in order to take some, we don't have room for, there are 2,000 whales in our fridges. Okay, 2,000 whales. We do not have space for 2,000 whales if we're taking the whole body. Not by any stretch of the imagination. And so this is a great way for us to be able to take tiny samples which can be used for genetic research and feed into understanding populations of whales in Irish waters. So this is a paper that came out in 2004, which drew on the collections that we hold to look at the DNA of species being stranded around Ireland, to help him, this is for a particular species, the white-beaked dolphin, and looking at the implications for conservation and climate-driven issues. This feeds into practical on-the-ground, boots-on-the-ground conservation at the end of the process. So there's a complete link between a stranded whale, citizen scientists, organisations like the Irish Whale and Dolphin Group, the museum, the research community, and that getting published and feeding back into people doing the boots on the ground work at the other end of this whole thing. And that feeds into legislation, it feeds into all sorts of things. Fundamentally important. This is just one example. I think there were 5 papers published on the genetic tissue bank last year, and there'll be more papers. There will be more coming out this year or next year, because we're still taking new specimens in all the time, and we're still providing access to researchers on a regular basis. This is Sean Dubay. He was doing a really cool piece of work on atmospheric pollutants, so looking at soot in the feathers of birds. He came up with a really cool system using the box and the white feathers on birds to be able to look at birds from different localities, different periods of time, to see how the soot loading in the feathers have changed. And it's interesting that after clean air laws come in, surprise, surprise, the birds get cleaner. So there are things like that which are going on. It's actually finding now that you're getting more birds with more pollution in their feathers from rural environments than in cities, because the way in which pollution in the air has changed, because we mostly focus on controlling in cities. So I think for me the takeaway is that the natural history collections we have help to document the responses to climate change, responses to changing agricultural use, all sorts of things, agriculture, all of that kind of stuff. They're really fundamental. And they help to feed into our understanding of the environment to be able to predict changes. So this is a nice paper. This is actually quite an old one. This is from 2002. But even then, these are huge scale models looking at where you would expect species to be disappearing when you factor in changing climate. And this is in Mexico. So it means that if you know how species are currently distributed, what kind of parameters they need to live in, and you can project how that will change over time with changing climate, it means that you can get a better idea of where you need to be putting your areas of kind of protected areas, so that you can keep some of that habitat, some suitable habitat for those species into the future. And so, just to wrap things up, so museums have a duty to use the collections we have to address linked existential emergencies, climate change and biodiversity loss. It's what we constantly try to do. We never have the resources to do it properly, but we do our best. And if anyone has any questions, ask away.